For a wonderful introduction. I'm grateful to be part of the Trust Global and today's webinar. I hope you all can see my screen.
Just check if it is in presenter mode in PPT. At the bottom of the screen, you have this presenter mode. Go down. See, there is something called comments, and there are some images, right? There you have this presenter mode. Now just go down to the extreme right of the screen in your PPT. You have zoom out, zoom in button, right? Left of that, you have one icon.
I can't find any.
Okay, no, share your screen. Share your screen, I'll tell you. 1 second, guys, please hold on. Just share your screen. Let me just fix this for you. Yeah, it's done, Ashni. You can start. Yeah.
Okay, so today's topic of discussion is sampling. So to start with, let us start with what is. When we do, for example, when we start a research project or when we start a study, very first thing what we do is we set the aims and objectives for the study. Okay, so for example, let's say our study topic, for example. So I'm from Chennai. So if a student comes to me and we are doing a study and say the topic is to find the proportion or a percentage of children that are anemic in South Chennai. So this is the study research topic, okay, a cross-sectional study. How do I begin this study? So when I say South Chennai, it's a huge population in itself. There are a lot of streets, there are a lot of people coming from different background, community, etc., etc. So to design a study, after setting an aim, I will set an objective and then calculate a sample size to see how much is an appropriate number of people should be incorporated in my study, so that my study will give the desired answer at the end and when we conclude it. Okay, so when we say South Chennai, it is a very huge population with different types of people and different types of cultures. So what we do is, we can't go to every street and every lane of South Chennai and collect the sample. Provided we can do it, but still it is time consuming, it is not cost effective, etc., etc. There will be a lot of constraint when we see a medical data. So it's always preferably good to have a sample rather than concentrating on a huge population. That's where sampling is really helpful for us. So as we deal with the medical topics, we have constraint in obstacles in collecting data, following up a patient, giving treatments which is expensive. When we have all these obstacles, what we do is a larger population, we will cut them in, and we will randomize or not randomizely, we will take the sample which will represent that population and start our study with, which will be easier for us and which will also fetch the same result as a population study will do. Okay, so what is opposite of sampling? Opposite of sampling is a complete enumeration. Complete enumeration is nothing but usually that happens in India, that is census, or else a population-based study wherein, for example, you say immunization study. So in this cases, what happens is we will not go into one particular street or randomly select a few people. What we do is we will take the data of entire population. That is called as complete enumeration or census, without leaving one subject or one person individual in the target population. Okay, now how do we do this sample? What kind of sample if we take, it will be the exact enumeration of the population and we will get the proper result. Say if I want to develop a app which helps me manage my manage my menstrual cycle, or else manage my diabetic to go for an insulin dates or whatever it is. If I have to develop an app, what I do is I have to see the different people, different individual who are choosing that sample, or different people who will use that app, rather than me and my team sitting and developing a app and which will be convenient for me or my team. It is not like that. I have to go in field and have to see what are different kind of population that will be using my app. If I'll develop a product, say A, what are the different kind of people? What is their educational background, occupation, their understanding level to use that app, so that my app will be fetching desirable profit at the end of the day. When it is coming into the market after the fourth phase of trial. To do this, what we have to do is at the very beginning, it is very important to choose a right sample from which is a representative of the population, so that we will get a desired answer. So sample will be divided into two types, that is random sampling and non-random sampling. Random sampling is also called as probability sampling. Why? Because probability sampling has got its own rules and principles which we have to satisfy, only then it will fit into a study. Whereas non-random sampling or non-probability sampling is very practical, where we can use it, all medicos can use it very easily or conveniently, because it doesn't have much of rules or much of principles. It do have principle, but it can be used practically, whereas probability sampling has particular principles which cannot be, most of the time, which cannot be brought into practical use when we are dealing with a very smaller sample size. So types of sampling are random sampling and non-random sampling. We will start with the random sampling. Random sampling is a method of selecting a sample in which each member of a population has known chance of being selected. In the sense, there should be an equal representation of the population. Whatever sample you will be going to if you have to check a IQ of a student in a medical class for first year MBBS, there will be 150 to 160 students in a class. You have to divide them into different groups for a certain program or certain practicals, wherein you have to check their IQ levels. At that point of time, if it is difficult for you to calculate it on 160 students, if you feel it is time consuming, you can take sample out of their class and then do a study on them to check what is their IQ level, provided all students should be samples should be equal representative of the population. In the sense, for example, if you only choose students from first row, what happens is, if all the students in first rows are very good students with the good understanding level, your sample will contain the population who are really good. So it will be biased that in your population there will be no students who are having or who are having lesser IQ or who are having poor understanding. So there is a selection bias happening there. So random sampling are of there are many types of random sampling, provided these are the major types of random sampling. That is simple random sampling, systematic random sampling, stratified random sampling, cluster random sampling, and multi-stage random sampling. Okay, so we'll start with simple random sampling. Simple random sampling is a very basic type of sampling method that we usually we start with. So all this, as I said, these type of sampling method, what happens is simple random sampling is a start, but when you go ahead like systematic, stratified, cluster, or multi-stage, these will be combination, or these will be having the sampling method which you learn priorly. For example, when you go to systematic, you will include simple random sampling and only then you can do a systematic. Again, when you go to stratified, it is same, simple systematic will be included in stratified. So if your base is good, then remaining all the methods are like inclusion of the previous one. Okay. Coming to simple random sampling. Simple random sampling is a method of selecting a sample from a population by giving all the members an equal chance of entering the sample. As I said priorly, if we will take the same measure of example of IQ testing, in a class of 150, it is always better you to, even though you're doing randomization, it should be equal representation of all the benches of the class, horizontally, vertically. Keeping in mind all this condition, you should draw your sample so that it will be an equal representation rather than going into biased selection. So simple random method will have, selection method is of two types, that is lottery method and random number table. These are the random number table. How do you use it? So if, for example, you have say you have put chits according to their registration number, roll number, or based on randomly, say phone numbers or any number. So I will consider to be you have put sheets based on your roll numbers. Okay, if you have put sheets based on roll number, your number, say if it starts from 10 to 21 or 12 to 40, any number what you have put, you have to go to this random number and start with first two numbers. Okay, first two numbers, or else you can start with last two numbers. That entirely depends on your wish. So if I will go to this, and if my number is between 12 to say 45, I will start with this random number table where I can see 12. So if I will start with this last two numbers, I have to continue. Only this last two numbers. I can't jump into first two numbers at the second call. Okay, so I will randomly pick numbers from this, or else you can do one thing is that directly take the random number table and then choose numbers like 64, 77. So you can call out that number and those students will be included in your study. Method of selecting a random sample from a population by giving all the members an equal chance of, okay, that is done. Ideal for homogeneous population, all members get equal chance. Estimates are easy to calculate. Difficult in constructing a sampling frame. Okay, so to simplify this, simple random sampling is having a very big demerit when it comes to practical use, because it will always suggest to have an equal number of population or identical population, that is homogeneous population. Wherein if you'll see in practical use, it is very difficult to get a homogeneous or identical population. Rather, we usually get a heterogeneous population only. It is or in unequal population, that kind of target we will be having. It is very difficult to have a matched or matched study or equal number of people with equal same characteristic, etc., etc. So That is one of the demerits, but to do a simple random sampling, this is the principle, or this is the rule which we have to see. And all members get an equal chance. So if you are having 150 people, all 150 of them should have an equal chance of appearing in the study. Nobody should miss out the chance of getting into the study, even though it is a probability then. Difficult in constructing sample frame. See, to read this, to understand random sampling is very easy because it's a very simple technique. But when we do it practically, it is difficult for us to construct a sampling frame wherein there is a heterogeneous population which has all the people are having equal representation coming into the study, and then following them up, calculating sample size, everything is a difficult process. Next, coming to the second method, that is systematic random sampling. What is systematic random sampling? In simple random sampling, we use the lottery method and we will pick this population into our study. But in systematic random sampling, what we do is first group. Okay, we will have, for example, say our population is say 100. Okay. Now our sample size, what we are expecting is 10. Okay, say whatever we are expecting is 10. In systematic random sampling, what we do is this first group, okay, first individual in the study will be randomly selected by simple random sampling method. But consecutive members who will be entering into the study will be depending on the kernel, like k, what we are calculating. That is, k is nothing but capital N divided by small n. That is, your population divided by your sample size will give the k. So for example, here my total population is 12, that is capital N is 12, and my sample is 4 people. Okay. So my me choosing a person into my study, first person will be randomly chosen. Okay, now say in this study only I have chosen second person. Okay, second person into the study. So next person who will be coming into my study is not random. It depends on the k that you calculate. The kth person will be entering into my study. So that is 5th person. Then comes 8, then comes 11. It goes on like that. So randomization will be done for the first individual rather than like in simple random for all the individual. Okay, so practically simple random sampling and systematic random sampling are the complex sampling method to use in a practical life, because these demand, first thing is to have a homogeneous population, second thing is to have an equal representation, third thing is to have a larger sample size, which we will not see ideally in the medical data, or in a scenario where you are having data like dealing with oncology, autistic kids, pediatrics, all this you will have a tough time to collect the data, especially microbiologists. You guys will have a really tough time to collect the data beyond 20-25 samples sometimes. So at this point of time, it is very difficult to work with the sampling techniques like this, systematic and simple. Next, coming to stratified random sampling. So now, what is stratified random sampling? How it is different from systematic and simple random sampling? Okay. So in simple random sampling, we use a lottery method and we choose a sample. Whereas in systematic, first person will be randomly chosen, and then consecutive person will be the kth person in the study that you are going to calculate. But in stratified, what we do is we will categorize the population based on our study objective. Like if you are giving a treatment to a patient, say treatment A and treatment B, which will act differently on say gender, male and female. Okay, treatment A will be acting differently on males, differently on females. Or else if you are in a trial wherein you have to see like women, in general, and you have to see if a woman is pregnant, how this drug is going to affect her. If a woman is normal, or if woman is having PCOD, or a woman with different categories, how the treatment or a tablet that you are working on, designing on, how it works on a different categories of women. If you have to check this, what you are supposed to do is, at this point of time, stratified random sampling is the best designing method that you can use, because this allows you to design the study in such a way that, based on the stratas or quotas or categories what you are expecting to study, it will help you to categorize your data in such a way, and then get the sample in that. So for example, a subsample is drawn from each strata by using any random sampling technique. So first what you are doing is, you will be designing the data, designing the study, and you will be doing different categories as you wish, like according to your study design, like male, females of different categories, whatever it is, you are going to do that. And then, using simple random sampling or systematic random sampling, whichever you prefer. Ideally, generally people use simple random sampling, and then they will draw samples to the subcategories, that is males, females, how many number of people. Subcategory, they will we can enroll the patients. Why stratification? To get representation for all the subgroups, to get more precise estimate, to draw the better conclusion on specified population, on specified age group. We are using this type of stratification, so that it will help us to get a on to the point, to the target bullseye results. Next, a matter of interest is cluster random sampling. So when we already have stratified random sampling, simple and systematic, what is the point of having a cluster random sampling? So when you do a study, what happens is you deal with different types of population. Okay, at certain point, like basically cluster random sampling, ideally most of the time, community medicine students, or study based on community medicine or health department, health ministry, they usually use this cluster random sampling, multi-stage random sampling. Why? Because people who deal with larger sample size can go for cluster random sampling. Because here what we do is, in stratified random sampling, you stratify based on, like, say for 100 patients or 200 patients, you stratify them based on their age, gender, or any specified category that is there in your objective, that is there in the mind, you will do that. But cluster random sampling is not like that. Cluster random sampling is a target for the entire population. Say you want to know 5-year immunization, how is it working for kids? Immunization, vaccine, how many kids are able access to the vaccines in India? What is the improvement? All this kind of study, or all this kind of objectives needs cluster random sampling. When you see vaccine access, you can't only see on India vaccine access of for pediatrics or infants or children in India up to age of 5 years. If you will see, you can't just go and see Chennai, Andhra Pradesh, or Mumbai, not like that. You have to go and see the entire India. All the states should be covered. Okay, if you say states, next comes district, taluks, villages. There are so many aspects wherein you have to go in and search, like get the data and then conclude the result. Okay, this much percent of infants or children, they don't have access. This much percent of infant have access, and there is one more community called as tribal community where you don't have access. There's so many aspects which will come before designing a cluster random sampling. So first what we do in cluster random sampling is, using simple random sampling, we will put chits, or else using lottery method, or else using random table, or else computer generated numbers. Anything is fine. Randomly what we do is we will choose the states. If for my example I have taken vaccine access for children less than or up to 5 years. So what I do is I will take put the chits, or I will take randomly numbers. I'll choose the numbers out of that chit. I will choose 10 states. Okay, from 10 states I will again randomly, either stratified or a simple random, I will use it, and then I will choose the districts out of that 10 states what I have got. Next what I will do is from that districts I will subcategorize into taluk. From taluk, I will use stratified random sampling and then subcategorize into villages, and then go and collect my sample from that 10 lanes, or that 10 colonies, or whatever it is. So that's how cluster random sampling works. The population is first divided into a number of clusters like slums, locality, family, school, etc. In my example, I have given you an example of states. Okay, a simple random sampling of cluster is selected, and all the elementary units belonging to the selected clusters are studied. So in cluster random sampling, what we do is we will not go beyond 3 lines, like a block design wherein 3 categories we do, and from that we will choose we will find the final population. Okay, that is one thing. In cluster random sampling, what happens is administrative convenience will not be there for us. It depends on randomly chosen whatever state, however it is, you have to go and administer it. It is it is cost effective provided you are funded so much, or else it is not cost effective. It is feasible provided whatever randomly chosen states or lanes is they are ready to respond, they are ready to cooperate with you, then it is fine. Or else again there will be a selection bias, non-responsive bias, there will be 100 things which will be obstacle to you. Okay, so in cluster random sampling we will stop with the third category. We will not go beyond that. Multi-stage random sampling is next method which is connected to cluster random sampling, because there will be a certain limitation for cluster random sampling. That's a reason multi-stage sampling is designed. Multi-stage stage sampling is sampling represents a more complicated form of cluster sampling, in which larger clusters are further subdivided into smaller, more targeted grouping from the purpose of surveying. So here what we do is it is exactly same like a cluster random sampling, but in multi-stage random sampling we have this liberty of subdividing your population, or subdividing your district, state, taluk, household, whatever it is, into a different categories until and unless it is a kind of convenient sampling for you. Okay, you don't have restriction like a 30 samples should be collected, or it shouldn't go beyond third categorization, third layering, nothing like that. In multi-stage sampling, you can do n number of stages, like tree to branches to leaves, like that. You can go down and down and make it according to your convenience, which cluster random sampling will not provide you. So these are the major probability sampling that we see, that is simple random sampling, systematic random sampling, stratified random sampling, cluster random sampling, and multi-stage random sampling. Okay, so what happens with this random sampling is, first of all, to design a sampling frame is very difficult with these things. Sampling frame means it is a listing of items that make up the population. Okay, sampling frame is nothing but it is a list of, before starting into the study you will decide what exact will be your target population, which will give you the following result. So that is the sampling frame. So frames can be anything: direction, maps, lists of household, village, taluk, anything, gender. You know, if you will go for a school, you know, if you are doing a study on disabled kids and you want to study that population, you will know in my study population, the South Chennai, there are 10 schools, 10 schools which is focusing on disabled kids, and out of that so many females and so many males. So you know, you will be knowing like what will be our population and all. So there's a high chances of you ending up with the bias when you use a random sampling in probability sampling in medical setup, because it's very easy, as it demands equal representation of a population. What happens is easily sampling error will occur. There is no doubt in that. Some, you know, if you should be that precise when you do your day study. Second thing will be you know, selection bias, non-responsive bias, coverage bias. There will be so many bias that will occur when you do the study, and at end of the data you will get a false result which is not actual result. So that's the reason, that's a time when researchers came up with this non-random sampling methodology, so that it will be feasible for all of us to use design our study in a practical situation and then go about with the study, so that there will be no error, no sampling error, and non-sampling error will be reduced, and bias can be controlled, and confounding effect will not be there. These are the reasons non-random sampling methods are in picture and widely used in medical science. So non-random sampling is a method of selecting a sample in which some members of the population have a greater but unknown chance than the others of selection. So this is nothing but there is no equal representation of the data. You will do the study with based on your convenience. Yeah, there are some you know, constraints or measure principles which you have to follow even in non-random sampling, but there is nothing like it should be equal representation, it should be homogeneous and all. Non-random sampling it will work fine with heterogeneous population, with you know, in your convenience you can sit wherever you are and collect the data, which you will see in everyday OPD, or else whoever can get you a data, you can use that data and you can start your study with. So non-random sampling are of different kind. Majorly there are snowball sampling, purposive sampling, quota sampling. Okay, so purposive sampling is also called as convenient sampling. So what is snowball sampling? To study a small group when members of the population are difficult to locate, first respondent tells you about the other eligible individual, who in turn tells you about the others. Okay, so snowball sampling is nothing but if your study objective is to understand the you know, say you are doing a study on transgender, MSM, or on drug, you know, youth who are under influence of drug. All this population are rare population. You cannot get, you know, if you will calculate it to be 100 or 200, you cannot find them. So when you are doing a transgender study, it is important for you to go to a center where you know, a rehabilitation center, community center, where they are located, or when they come for treatment, or else you have to find one transgender in toll or wherever you find, and through her or him you have to find one more person and then go and take their interview. system. So this sampling design, the calculation of sample size is also very different. Sometimes the calculation of sample size will be kept as operational for this, because you certainly don't know how many transgender you will be you can get in contact with, and how are they going to respond. Okay, in this scenario, it's a major problem will be of response. There's a response bias. So the studies cannot be done in 1 or 2 months or 6 months. It is a longer procedure wherein you will wait until you get a specified population which is statistically and clinically feasible, and then you will go about with the further analysis. You can't rush with this sampling method, and this sampling method is used only for the rarer population wherein you can't find them easily, like drug addicts, transgender, criminals, or if you are doing study on rapists, if you are doing study on transgenders, all these are the rarer population and difficult, tough population whom you can't, they will not give you a response immediately as as a normal population. Okay, this is where you have to use the snowball sampling. Purposive sampling. Purposive sampling is a very common sampling method which is widely used in medical science. Okay, every person, every student, basically in medical college setup, and professors, whichever study they do, either it will be designed as a convenient sampling. Most of the time, will be a convenient sampling, or else a purposive sampling. So what is this convenient sampling? Convenient sampling is nothing but in your convenience. Okay, larger population, you cannot, there will be a chance that most of the people will not be part of your study who actually should be part of your study. There will be a selection bias, obviously, but you will sit in your OPD, or you will go to a place of your convenience, and you will take or collect the data. Okay, it can be a self-administered questionnaire. It can go like any study, like if you want to do a study on autistic kids, if you want to do a study on for example, any hospital-based study, if you do, it will be a convenient sampling. So they say whoever comes to our OPD, we will be doing study on them. So that's how it goes. Sample size calculation for convenient sampling are of two methods. So convenient sampling, one thing we can do is we can put Dobson formula, and we can calculate and say, okay, this will be the sample, if it is a prevalence study, we will say, okay, this is the sample size, you have to calculate and collect these many, no matter what. Okay, but the second method is period sampling we do. What is period sampling? Period sampling is nothing but, say I am here for 1 year. Okay, my study is for 1 year, and I want to know the if I'm doing a study and my study is on my study setting is my hospital OPD. Whoever comes to my OPD for next 1 year, I'm not going to leave one single patient, and I'm going to collect data from everyone who will be entering the OPD with the study objective condition inclusion criteria set by me. Okay, it will not stop for 10 people, 15 people, or 20 people. It will be like given a 1 year or 2 years of time, whoever is going to coming to the place of study setting will be enrolled into the study, and then the study will be proceeding. Okay, this is the two method of calculating sample size for purposive sampling. Okay, those being interviewed fit a specific purpose or description that was necessary to conduct a study. So basically, inclusion and exclusion criteria are very important when you do a purposive sampling, and it is also important for you to think, like if there is a selection bias happening in your study, or if there are any confounding factor which you feel may be there when you do a study, you should take care of all that and then do the study. For example, if you want to know. Okay, so your study is to see in your convenience. If you want to study a patient, say, we will take the example of anemia only. So if whichever woman is coming to your hospital with the complaint of iron deficiency or anything, you are going to study that woman or that population. You can't say that, okay, uh, my result. So proportion of people who are anemic in the proportion of women who are anemic is 50%, 20%. You can't conclude like that. You have to be very specific, telling that provided the condition was my hospital OPD. Second condition was the people coming to my OPD are mostly Christians, mostly Muslims, like that. What is the specific religion or background or culture they are in? And you should be very precise while reporting your study, that provided so and so condition, my study shows that 70% of the women are anemic in the Krompet district, or else in Chengalpet district, anything like that. You can't say that directly 70% of the women are anemic in Chennai. You can't conclude like that. You should be very specific when you do a purposive sampling. Provided so and condition, a person is anemic, something like that. So you should provide the condition, what you have done, study, circumstances under which your study has been done, and what are the things which you have not been assessed. Okay, next coming is quota sampling. So quota sampling is nothing but it is a non-probability way of doing stratified sampling. Okay, quota sampling, what we do is, a researcher will divide the data into certain categories like gender, occupation, anything based on his objective. He will divide the complete population into a category, and then consider or enroll the patients into, or subject into the study. So say I want to see the number of I want to know a number of youth who smoke, what kind of brand of cigarettes they smoke. Okay, if I want, my study interest is to know which age group of person smokes which brand of cigarette in which city, then what I do is, if for me doing cluster random sampling is very difficult in practical, so I will take a non-probability sampling and put a quota sampling, wherein what I do is I will divide the group into ages like 19 to 25, 25 to 30, or 30 to 50, whatever categories I divide, and then I will enroll the, I will categorize the them. I will take, randomly I will choose in my convenience, not using any simple random sampling or stratified. And according to my convenience, I will choose certain cities, and from there I will choose randomly patients to my study, youths to my study, based on my convenience, not based on any particular random sampling like simple or stratified. Okay, this is how quota sampling is done. Basically, to understand, your study objective is on specified population, specified age category, gender, occupation, or anything. It can be anything, males, females. So to understand this, precise the result if you want to get, or if you want to study on a particular group, then you can use a quota sampling. So the sampling technique begins by forming quota. Quota is nothing but category or strata, anything similar to the stratified sampling, but the sampling will be within each strata is done by non-random method, that is not using any simple or stratified method. Okay, so here we come to end of Non-random sampling. So random sampling, to brush it up, we understood what is simple, stratified, systematic, cluster, multi-stage. In non-random sampling, we understood what is purposive sampling, what is snowball sampling, what is quota sampling, which are usually in use when we do a design or study. Next, coming to this one more category called as designs, or we say others, wherein especially in trials, when we do clinical trials, mostly we use this kind of experimental designs. That is, there are different kind of designs. That is block experimental design, Latin square design, and factorial designs. But today what we are seeing here is especially a block experimental design and factorial design, because the Latin square design is mostly used in agriculture, which is very specifically used in agriculture. So it is of here we are discussing about only block experimental design and factorial designs. Okay, so what is block experimental design? So as simple random sampling, here also block are nothing but different groups. Okay, block are nothing but different groups. What we do here in block random sampling is we will divide the population into different groups. That is, say we will take gender only. Okay, we will take males and females. Okay, and we are giving a treatment to males and females. We will say I will give in my trial I have 3 tablets, the treatment 1, treatment 2, and treatment 3. So what I do is for males I will give treatment 1, treatment 2, treatment 3, and for females also I will give treatment 1, treatment 2, and treatment 3. Okay, here what we do is, here comparison will be male treatment 1 with the treatment 2 and treatment 3. Females again comparison will be within treatment 1, treatment 2, treatment. So it will be whatever permutation combinations we will get, we will compare it within. Male to male treatments will be compared within males, female treatments will be compared within female treatments. So it is not intra comparison, it is inter comparison happening. So to understand how specific treatment or specific tablet work on male population alone, or how it works on female population alone, if you want to know an individual effect of a treatment, then you are using a block experimental design. In block experimental design, local control, principle of local control is used. What does that mean is, again, it should be a heterogeneous populations are not much of it's not much entertained. I will not say it is not used, but it is not much of we will not expect it to be a heterogeneous population. And different blocks are created according to researchers' convenience. There is nothing like you have to it should be equal representation, it should be based on any designing method, nothing like that. Blocks can be created based on the researcher's convenience. Presence of block study becomes easier. See, if you have a targeted objective and if you want to know one specific group of individual how a treatment or how a vaccine or anything is working, then this design is best designing method, so that you will specifically see interaction or whatever relationship you are causal relationship you want, desired causal relationship you are seeing, it will be within group, not intra group, intergroup. Okay, this is what is about the block experimental design. Next, coming to factorial design. In block experimental design, what we have done is we basically see what is the individual effect. Okay, individual effect of a person, that is treatment A, and males to males, females to females. But factorial design is helpful when if a researcher or if a trial is interested, a principal investigator is interested to see the interaction effect, or a company or a sponsor is interested to see the interaction effect, means A person, the study has to adapt factorial design method. The designs are used where the effect of more than one independent variable on the dependent variable is to be measured. Basically, causal effect. If something, because of something, something is caused. For example, if a person smokes, will he end up with cancer? If smoking is a reason for cancer, so this is called as causal effect. Causal effect is nothing but one variable is causing something, okay, is a reason for a person to develop something, okay. That is what is causal effect. So to check the causal effect, to check the interaction effect, no matter how many permutations and combinations are there, factorial design is the best method. So factorial designs are of two types, that is simple factorial design and complex factorial design, okay. In simple factorial design, what we do is we obey this two square method. Two square method in the sense, for example, say I am doing a study. Say my drug is a paracetamol. Paracetamol 500 mg and 650 mg. Two groups. Okay, I'm giving patient paracetamol 500 mg and 600 mg. Second variable of interest is I'm seeing paracetamol, a person who is given 500 mg, given empty stomach, what happens? Given full stomach, what happens? Again, paracetamol who has taken 650, when he is having full stomach, what happens? When he takes it empty stomach, what happens? So you are having here four interaction effect, or four relationships which you have to find. So this is the design for simple factorial design. Complex factorial design is what is 2 cube, we say 2 cube in the sense, first what we have done is paracetamol is a tablet where we have divided it into 500 mg and 600 mg. With the second variable will be person has taken it empty stomach, not empty stomach, 500 mg. 650 mg will be again divided into empty stomach and not empty stomach. Three complex design will be one more layer you will bring into the study. If a person has, if he is having any other, a person is diabetic, not diabetic. Person is who has taken 600 mg, again diabetic, not diabetic. So the interaction will be doubled here. So the relationship to find will be the design is having complexity because the relationship finding, the interaction level will be higher. That's the reason it is called complex factorial design. Basically, if you want to know the interaction between two drugs, interaction effect between two drugs, we will use factorial design. Coming to a very important aspect before, with all this sampling technique, experimental design, all these techniques are, with the error. Okay, so that's the reason we say statistics. We don't go with mathematics. Statistics and mathematics both deal with number, but they deal with number in their own way. Okay, what is beauty about statistics is it believes in uncertainty. Mathematics believes in certainty. Okay, statistics gives you opportunity to calculate your result or data with error. Okay, statistics never says that something is 100% perfect, but mathematics always says that things are 100% perfect. That's a reason in any medical science or data analysis, we don't use mathematics, we use statistics, because we set, when you can do a sample size calculation or whatever you do, you say 5% error, 10% error, with this margin of error I'm going to do my study. So you will pre-decide your error and then do your study. Say that at the end of the study, when I get my result, whatever result I will get may have or will have 5% error. With 5% error, I am telling that 70% of the population is anemic, 20% of them are diabetic, something like that. So you always speak in terms of, I'm not 100% sure, 95% of the time I will say this is right, or this is this much proportion of people are, anemic, something like that you speak. Okay, and that's the reason there is always there's one more entity called power. Even though 95% I'm sure my test whatever I've applied is 80% of the time it is stronger, it gives it detects the perfect answer. That's what is the power says. So with the combination of this, you will do a study which will give you a approximately same result as a population study, and you will publish your data or result or whatever it is. Okay, so what is sampling error and what is non-sampling error? Sampling error is nothing but when we say you're drawing a population from a when you draw a population from a sample, when you draw a sample from a population, I'm sorry, sample from a population, it should be an equal representation. All should get an equal chance of appearing into a data. Or when you do a study, it should be, you should see that older and younger generation can use that product or use that medicine or whatever it is. It shouldn't be biased to one age group, biased to one gender, or biased to one kind of population. So this sampling error is an error when this kind of selection bias or non-responsive bias will happen. That is the time when we say it is a sampling error. Another form of error is non-sampling error. Non-sampling error is nothing but error when the error that happens when you do a study. Okay, when you do, see, this is non-sampling error is also called as a human error. Okay, so what happens in non-sampling error is first thing that we do is when you do a questionnaire based study, if it is a self-administered questionnaire, or sometimes people will not answer your survey properly, or else if it is if it's a follow-up study, people will not come up for follow-up, or else the third thing is that if you are administering a questionnaire, and if a person is not eligible to answer your question, his caretaker, mother, parent, or somebody will answer your question, and probably that answer may not be right. So there are there are many types of errors that may happen. Measurement error is nothing but, see, when you take you say there is Spigmo BP checking machine, which is manual and which is this one digital. So you want to know the agreement between whether manual is good or digital is good. What we do is we will take repeated measurements with both the devices for same patient and see how is the readings given to check the agreement. If one study, if they say there are 150, 200, 500 patients, the study will be done by different person. The data will be collected by different person, and this different person, every person are different. Every person, the way they do study is different. So whatever measurement you take, sometimes you write it differently, or you calculate it differently, or you measure when you do a reading. There is a possibility that you have not made a person rest and then take his BP. There's 100 reasons because of which the measurement what you have considered is a wrong measurement. So that's a measurement bias or a measurement error. So all this non-sampling error are the human errors that is being made, and sampling error are natural errors, which cannot be corrected. But non-sampling errors are the errors which can be corrected because it is who we are making. Sampling error are natural. Okay, sometimes certain times we can correct it, but certain times there is a possibility that we cannot correct it. It happens. So there is a scope for that. Okay, but non-sampling errors are not entertained most of the time. So these are the two types of error which you have to keep in mind before doing any type of study. Yeah, so this is the methods to minimize the error. A very simple method is to increase the sample size. A better sampling technique. So probability sampling is always considered to be a better sampling technique, but which is not feasible always. So when you do a non-probability sampling method also, there are different ways which will make your study a better study, even though you are doing a non-probability sampling with with lesser error, with you can prevent doing so many errors, provided you talk to a proper expert who will guide your study in a better way, and you can get a very appropriate result, approximate result. Okay. Non-sampling error, ensure confidentiality. It's like if you're doing a clinical trial, double folding, single folding, double folding, all this comes into picture. And design a better questionnaire. So questionnaire should be always a proper representation of your study. It shouldn't be having unnecessary question which is not required, which will not target your objective, random questions. It should be a proper questionnaire which will undergo reliability, validity testing, test retest methods, and then which will be given to a population, so that we will minimize the error. Send reminders to survey participants if you are doing a follow-up study or on-call surveys. Avoid short data collection period. Okay, so these are the methods to minimize error. Use incentives if you are funded, or if you have that much provisions provided. Train the interviewer. So if you are sending appointed 3 to 4 trainers to do one study, all the 3 should be it will be trained well, so that they will usually the approach of asking question, making a participant comfortable and feel confident to give any type of information to the surveyor. It is very important how you present, or how you talk to the patient, or how you make him feel comfortable. So the trainer, you should train the interviewer well, so that he will get a proper result what you're expecting. Okay. So yeah. This is it about sampling techniques and experimental design. Yeah, thank you.
Thanks, Ashman, for your wonderful presentation. So, audience, if you have any questions, you can ask. It was very nice, because these are simple things that you should know when you are working in a life science industry, I feel. See, statistics is a huge. You might think it was just 20 slides and how no, but this is very important in the industry. So let's have some questions from Dr. Surya. Please go ahead.
Hi, that's a good presentation actually, and my question is regarding sample size and sampling error. So
Okay.
like given to exactly the same studies and same sampling methods, same population, the study with a larger sample size will have less sampling process error compared to study with small sample size. So that's what obviously you said, to increase the sample size in how to reduce that. So there are studies where the sample size are very less. So how to overcome this, and what is the ideal method for it? So
Ashmi, I think you got the question. You can answer it.
Yes. Hi, Surya. It's a good question. So there are many scenarios where you don't, as I said, microbiology or oncology, ophthalmology. These are the areas which we usually see people struggling with the sample size. Okay, so it is there are n number of methods when you calculate a sample size. So if you feel that your study can't go beyond 20, 10, 15 or 30 samples, that's the time when you have to usually it's obvious that you will use a non-probability sampling method, provided you should talk to a proper expert or a statistician or epidemiologist who can guide you with the sample size. So when you calculate a sample size, there is something called power of the study. There is something called significance, confidence interval, and margin of error. Plus, there is something called non-responsive rate calculation. There are so many different types of calculation, which we will with the smaller sample size, you can do a appropriate, better study. Provided everything lies here in the sample size calculation, because if you will tell the statistician or whoever is helping you, community medicine professor or whoever it is who is helping you, if you will tell them your exact study and what is the merits and what is the limitation of your study, they will definitely help you with the sample size, which will help you with the doing a better study. And second thing what you have to do is you have to check is there any confounding variables. So if there are any confounding variables, you have to, if possible, see certain confounding variables we cannot identify, but certain confounding variables it is very obvious that we can figure it out. If you will let all this if you will tell all this, or if you will figure it out, then it is possible to do a better study with a lesser sample size.
Thank you, Ashni. There is one more question from Mr. Amit.
Yes.
Yeah, please go ahead, Mr. Amit.
Yeah. This was a very good presentation. Thank you for the presentation, ma'am. My question is, I have read many research articles where in methodology section they write that they have interventional study.
Sorry?
Having two groups. Hello.
Yeah, go ahead. Yeah, go ahead, Amit.
Intervention.
Yeah, intervention study.
They mention in the methodology section that they have an interventional study with the two groups, and they randomly allocate the participant into the two interventional groups, one interventional and control group. Now, during the presentation, you told that in simple random sampling, we need to sampling frame first to randomly allocate the individuals into the different treatment groups. So how in practical sense does the randomization with allocation concealment is done in the clinical trial?
Yeah, so I'll answer this question. See, let's assume that it's a crossover trial, it's an interventional crossover trial. There are 100 participants which are going to use. So group A will be 50 and group B will be 50. So now I have to equally ensure that it is randomized. I cannot, I have already double blinded my study. So I have to ensure that everywhere it's double blinded. Randomization at the start level, it's double blinded. Then at the statistics analysis level, it's double blinded. Person who's taking the measurements, data collection, it is double blinded. So nobody is going to know what is happening. So that is one way of reducing the bias. But how you can randomize here is, for example, A, B, B, A. So this is simple randomization, lottery method. So then everybody will come to know first it is A, then it is B, then it is A, then it is B. So what I do is I create blocks. So first block is 2. Second block is 4. Third block is 8, 6. So that means permutations of 2 is what? 2 into 2. Permutation of 2 cube is what? 2 into 2 into 2, 8. So that means I have A, B, C, D. I have A, B, B, A. I have A, B, B, A. A, B. So I have various things that are jumbled together, and I take one and put it in one group, which even I won't know that whether I am going to put this person into the group, whether it is going to be A or whether it's going to be. So I use probability here to generate the samples. So I create blocks, block of 2, block of 4, block of 6, block of 8. This is how you do sampling in a clinical trial. In clinical trials, there are various ways of doing sampling. You have stratified methods, you have block methods, then you have stratified block methods, cluster samplings. Nowadays it is mostly they use a mixture of all those things to do. So it all depends on what is your inclusion criteria and exclusion criteria, how stringent you are, and how much blinding you are going to do, whether it is a double blind or an open blind or a triple blind study. Blinding is at what stage of the study, whether throughout the study you are going to blind the participants or you are going to unblind it at some stage. So all this matters when you do the randomization. I think Ashley, you can, if you want, you can add some more points if I have left out something.
No, that's what they do. They will create a random system, will generate the sequence for them, and then they will, it will come with the envelope to the first stage of treatment. Investigator, whoever is giving the treatment. That's how they do the sampling there. Usually they use a multi-stage sampling method wherein all the sampling techniques, like from as Sir said, simple to cluster is involved, and they do it because multi-stage sampling doesn't have much of principles or rules which you need to take in consideration.
Correct. Any other questions? Yeah, go ahead, ask.
My question is, you said we will create blocks either
Correct.
in multiples of 2. Whether these blocks are homogeneous means like stratified randomization, or
It depends on your
It
criteria.
depends. It can be heterogeneous also. It is not necessary. It should be homogeneous.
okay. And the number that AAB is randomly generated.
Yeah, that is system will generate the sequence.
Okay.
Correct.
Thank you.
So let me just show you how it looks usually. Just hold on. Let me share my screen. Just a quick thing I'll show you. So let's go to recent. Yeah. So I think you're able to see my screen.
No.
Now? Are you able to see it?
Loading.
Yeah,
I think it's loading.
Okay. So see, this okay. This is from a trial itself. So
I
see
think
this.
Here it has written you don't have access to this file.
It's not showing in your screen?
No.
No,
No, it is showing. I think it should be showing. Just hold on.
it just came like content can't be seen.
Oh, is it? It's my company content. I'm not sure why it is. Will be some security features the compliance team would have kept. Let's see. Upload a copy. I think now it should be visible. Invited to this meeting and edit. People can't access this file. Oh, I don't think it will be visible. What about now? Is it? Are you able to see?
No.
Okay. So I wanted to show you this randomization, what I told you. So I am not able to show it. I'm sorry.
Yeah, that would be great actually. So means we can actually have a visual impression how it actually performed during that.
So what I'll do is, anyways, since you wanted that, no, I will share one dummy file for this randomization for the same question which you asked for a clinical trial. There are 2 groups. I'll create one dummy file. I'll share it over the weekend with you guys in the WhatsApp group. That would be great. You can see that. It's a computer generated, software generated. So maybe 1 or 2 scenarios will use like 3 groups, 4 groups, 2-arm trial, 3-arm trial, 4-arm trial. How randomization is being done with the study title and everything. I'll share it with you. Don't worry about it. Sure.
Hello.
Yeah. Tell me.
Thank you. And can we include allocation concealment procedure in that also, so that?
I'll include it. No worries. Yeah.
Thank you.
Sure. For a learning curve, I'll include that. Yeah, sure. So any other questions?
Yes, I have one more.
Yeah, sure. Go ahead.
Like just as one of the problems of sampling is like lack of sample representatives. One of the common so like example, if the population under a study is heterogeneous and the sampling process is carried out in the absence of consideration of nature of the population. So it has been done in one of our studies as well. So just an tip or something how to avoid these things on.
See,
yes, I know for better planning that.
Yeah, one is in when we do a clinical trial or whatever it is. See, now it is all there are softwares which are AI driven, which can take you the literature. They can take you geographical representations of each population stratas. But in my opinion, the technology is evolving. So what I would suggest is you have to refer to the registries. See, for example, if you are doing a study on diabetes and you are including patients at the age group of 18 to 60 with insulin, without insulin, patient who have taken anti-diabetic drugs, this age group, so many complications. Every strata has to be having a set of population. So you have to go check in that whether they fit your study. Rather than looking at a center or let's assume that you are doing a study in Chennai in the hospital. So what we do, we just only concentrate on people who are coming to that hospital into the diabetic department. And we try to squeeze something out of that using our inclusion exclusion criteria and we fit the study. But when you plan a trial, it's not that how you choose a center. It doesn't matter. There are so many centers which can do the trial. But it is the planning. How do you attract your population? That is your subjects. How do you recruit? Do you have a plan in place for that? So it can be one proper advertisements can be given. Proper registries can be checked. EHR and EMR should be integrated in your clinical trials. Which trial? You tell me one trial where EMR and EHR is integrated. No, it doesn't happen. So the hospital or the site which you choose should be electronically driven. You cannot go choose the participants by looking at your MRD files. How many files you will read? You will definitely miss out on data. But if same thing, if it is driven electronically, you can bring that data into the system where you will have common data fields and you can automate it. So I tell age group 18 to 60 in this center, I'm only going to see patients with diabetes who has peripheral neuropathy, who has glove and stalk neuropathy, who has different types of tingling sensations, one in the fingers, in the cuff muscles. So who has sensations in the tongue. So all these qualify as people for peripheral neuropathy. So I am not going to look into other subjects at all. I'm going to screen exclusively for that criteria. So one center is dedicated for complications. One center is dedicated for. So you have to think like that. Rather than just enrolling diabetes patients and looking at their records and pulling the data and saying that, oh, this person is qualified, he fits the criteria. The criteria, this is what is called as trial enrichment. So technology should play a major role. One of the technologies is your EHR and EMR. The second one is biomarkers. So whatever biomarkers you are going to analyze as outcomes, my drug is going to reverse this biomarker. So let's assume that NF-kB levels are increased in multiple myeloma. So I am using a bortezomib as a drug to inhibit that NF-kB levels. That means I have to choose participants in such a way that they have so and so threshold for NF-kB levels. We have to design in such a way that the biomarkers are getting identified. So that will increase your study enrichment. So these are all things that comes under clinical trial enrichment criteria. So I feel it should be a multi-driven approach, not just a single center or a organizational approach. It should be the policies that needs to change. To enroll somebody in a trial, you have to have certain criteria to qualify that person as a participant in the trial, which should be laid down by the regulations itself. The sponsor should not decide it. The government should decide and tell. If you are doing an oncology trial, it should be done in a structured way like this. Then your trial enrichment is good. Then your quality of data will be good. Then you can show better efficacy for your drug. and that means your drug can even fail at the end. So it's a negative trial. We should welcome negative trial. So what happens halfway down when the trial becomes a little jittery, they stop the trial. They say our drug failed. So we have invested so many billions. We have failed this trial. We'll come out with another trial. Obviously, everything will be insured. The companies will get the money back. So that should not happen. So I'm just telling you how it can be in a broader perspective. In the future, it can be improved. Yeah.
Yes, sir.
So,
But
yeah.
like that you said that to check the registries, and also like I'll just give an example of what went wrong with our things. Like there is an village in Karnataka with active leprosy patients. So
Okay.
infection. So
Okay.
as per the recent registries of 2022 and health records of that Karnataka, those things, the set population with active infection is 110 to 115 people. So we took the sample size of 75 by a like simple random sampling. But so what happened is after starting of the study, we have there is no representatives of that sample. So we took like it like 25 samples, 25 subjects are not also not got. We didn't got.
You were not able to enroll, is it? You are not able to enroll that 75 subjects. So what was the reason you were not able to enroll?
25.
So what was the reason you were not able to enroll? Did the participants didn't come, or was there informed consent and patient information sheet, or the knowledge which you transferred them regarding the trial was not enough? Did they understand it? What was a major problem? Did you look back at it?
Yes, sir. The major problem is the infection. I mean, the rate of infection is absent.
Okay, so why do you want to restrict yourself to that particular village in Karnataka? You can select other villages in India as well, right? You could have made it multi-centric, right?
It's a small study, but
see, this is one example. If it's an academic or a corporate study, I mean industry study.
No, sir, it's an academic.
See, academic trials, that is always a minus point. Academic trials, where is the funding from? That's what institute, IITs, you call it, right? Investigator initiated trials. Investigator initiated trials should be supported by the colleges or the universities. But where do you get? You have to apply for grants. You will get some 5 lakhs or 6 lakhs. So that is going to be a bias in the investigator. That is no other way you can do it. Corporate trials usually don't face such problems nowadays. They are investing money, but I feel for academic trials, you have to have association with the corporate also and take some funding from them for helping the trials. Yeah. Yes, so last question, Amit, last question from your end. You have raised your hand, then we'll wind up.
No, that was for previous question.
Okay, fair enough. Yeah. So I think it was a good session from Ashni. Thank you, Ashni. We would like to see you again in the forum. Of course, you will be a part of this forum, so we can discuss something different next time where it will be helpful for both academic as well as industry peers. So the idea of this forum is to give equal knowledge to people who work in academics as well as in industry, where there is a common interjunction where everybody works. So we select topics in such a way that both the academics and the industry folks are benefited in what they listen. It's not just purely industry, it's not just purely academic. So thank you guys for your participation. Let's meet next week. Thank you very much. Good night.
Thank you, sir.